This is a tutorial for iProteinDB. I'll be showing how to search for post-translational modifications in Drosophila, as well as a few other features. So to start out, you're going to enter a list of genes or proteins with one search term per line. You can search by flybase IDs, gene symbols, and gene annotations. It's also possible to upload a file in this format. I'm just going to be using the sample terms. So after hitting submit, we're taken to this table where the first column shows the term that was searched as well as the gene that it was mapped to with the number of distinct protein isoforms. If we click on one of these gene IDs, it's going to show us some predicted orthologs over here, and then a table of the protein isoforms with their sequence length and number of observed modifications. To run a multi-sequence alignment of all the protein isoforms, you can click here. This will run for a minute, and then the results are shown. So now I'm going to click on one of these flybase IDs for a particular protein isoform, and this is going to take us to first a protein details tab for this particular protein. First we've got the sequence, where there's a key down here showing what each of these features are. So if it's highlighted in green, that's part of the sequence is in a domain. The amino acids in red are observed PTMs. Scrolling down, we also have the PTM sites in table form with links for more information, as well as more details for things like the propensity score and the cross-species alignment. Scrolling down, we have the domain section. So this is showing the start and stop position within the sequence, the name of the domain, the description, as well as a link out to the publication that this was from. Next is subcellular localizations. Then finally, kinase target predictions. And here, these are based on ScanSite 3. And if you want to see some orthologs for these particular genes, you can also click on the plus sign to be linked to DIOPT. So going back to the top, the next tab is abundance data. There's table format data for life cycle and embryogenesis, as well as a graph. If you hold your cursor over one of these time points, you can see what the exact intensity was at each. And finally, there's a cross-species orthologs tab. So these are showing orthologs in model organisms. We've got RefSeq IDs, NCBI IDs, as well as some more information like the DIOPT ortholog prediction score. Scrolling down, we have domains from each species, where within the sequence the domain is, the name and the description again, as well as, of course, the link out to the publication that was found in. Scrolling down, we have subcellular localizations for each species, and then we have the conserved sites with positions, a 5 amino acid window on either side of the site, whether or not there's an antibody available, and the average identity. And this table shows available antibodies provided by cell signaling technology with catalog numbers that link to their website. Next is human disease-related variants from UniProt, assuming there are any. In this case there weren't. And finally, there's a multiple sequence alignment for the model organisms. Clicking here, again, we'll run that, and we can see the results. It's using the same format as the protein details sequence view. And then if there's a particular region within one of the species that you're interested in, you can choose the organism here, and then the position that you want to highlight so that you can easily see where that part of the sequence was aligned. And that covers all the basics. If you still have questions, our contact information is available here as well as a bug report. Thanks for watching.